review questions at the beginning. I'm going to move through vitamin A, and then after the break period, I'll spend some time reviewing some of lectures 1 through 6. So after break, if you're not interested in kind of lectures 1 through 6, feel free to leave. You won't hurt my feelings or anything. But I thought this would be a good opportunity to do some throwback to get ready for the exams, since it is cumulative, and touch base on some of the slides and particular concepts and topics that I thought were important in the first lectures from the first section. But first, let's go through today's lecture, which is on vitamin A. So we'll start by going through the chemistry, some of the sources of vitamin A, its route of absorption and transport, biochemical activities, and biological functions. So vitamin A was discovered as a fat-soluble factor present in animal fats and oils that was important for growth. And then it was later discovered that this was also present, at least in some forms, in plants, mainly as carotenoids, which are precursors for vitamin A. So collectively, vitamin A refers to many compounds that exhibit this activity, the biological activity of retinol. So this is a wide variety of compounds. Some of them are synthetic and some found in nature. And vitamin A plays a wide variety of roles in the body, particularly in vision, and this was where we'll focus our attention, but also plays important roles in immune function and reproduction, cell communication, and cell growth and differentiation. And so this is showing the basic structure of a retinoid and the active form of vitamin A. So this is all trans retinol, and so you can see some of the primary features and why it is a fat-soluble vitamin. Just given the structure, it looks like it would be a fat-soluble vitamin. It has this beta ion ring structure, this polyunsaturated chain spacer, and then a polar head group. Okay? And these polar head groups can be at various oxidation or reduction states, which will give different properties to the molecule. So as we go towards more oxidized, we go from retinol to retinal. Okay? So you'll see that the polar head group here is becoming more oxidized. And then even more oxidized, now we have a carboxylic acid group, and we have all trans retinoic acid. Okay, so we have retinol, the alcohol, retinal, an aldehyde, and then retinoic acid being the acid, right? The carboxylic acid group. And these are going to have different activities. Primarily, we'll focus on some of the retinals, which are going to be important as chromophores in our eyes, and then all trans retinoic acid, which is going to be important through its regulation of gene expression. Okay, so not surprisingly, the most famous source of vitamin A is carrots. We all probably have heard eating carrots is good for our eyes. But it can also be found in many other leafy vegetables, and other fruits and vegetables, cantaloupes, tomatoes, fruits. To some extent, it's present in good amounts in liver and fish oils, to some extent in milk and eggs, and then of course we're in the age of fortified cereals, and so we can get most of our vitamins from fortification. And then there are a variety of different dietary supplements on the market with retinol present in different forms. So those would be retinol acetate, retinol palmitate, retinol, or beta-carotene, which is the form that we get from plants. And these can all be converted into retinol and active, biologically active forms of the retinoid. Okay, so in terms of in our diet, retinol is gonna come in, vitamin A is gonna come in two primary forms. We'll be getting it as retinol esters, and these are going to mostly come from animal sources. And so here we can see this would be our retinoid molecule, and then it's esterified with a fatty acid. So this R group here would be a fatty acyl chain, right? So it's an esterified form of retinoid. Okay, and this is a storage form of retinoids. So this will mainly be coming from animal sources. The other primary source is coming from plants, like carrots. So this is all trans beta carotene. And you can see that this looks kind of like two retinoids stuck together, right? If you chop this in half, it would give you two retinoids. Okay, and so this can be, sometimes this is called like a pre-vitamin A, because it can be metabolized into the active form by cleavage. Okay? So this is mostly from plants. Well, this is completely from plants, beta carotene, and then these retinol esters mostly from animal sources. So in terms of the absorption of these different molecules, retinol esters is going to require some metabolism before we can absorb it into our enterocytes. Okay, so similar to other things like cholesterol esters, you remember, it's going to need to remove the fatty acid that it's esterified to. Okay, and so this can be done by carboxyl ester lipase. This is the lipase secreted from the pancreas, which also metabolizes cholesterol esters, which we talked about previously. And it can also be hydrolyzed by retinol ester hydrolase. And this is a hydrolase that's expressed on the enterocyte brush border. And so both of these enzymes can contribute to the removal of that fatty acid esterified group. Okay, so it's converting it from a retinol ester into retinol, the alcohol form, plus our fatty acid. Okay? So it's just hydrolyzing that bond there. So this is what would happen, right, for things like triglyceride or cholesterol esters, also in our diet. So very similar. So then these free molecules can then be taken up by our enterocytes. So retinol will diffuse into the enterocyte and will be bound by a cellular retinol binding protein, abbreviated CRBP. And our fatty acid can be taken up by other routes like CD36 transporter, right? Beta carotene seems to be actively transported into the enterocyte as that entire molecule. So this whole molecule is actively transported into the enterocyte. Okay, so there's no pre-metabolism of this that's required for absorption into the enterocyte. However, after being absorbed into the enterocyte, this can be cleaved. So again, this kind of looks like two retinoids attached to each other, and indeed, following cleavage by this beta-carotene monooxygenase, BCM01, it's present in two molecules of all-trans retinal. Okay? So that's kind of the aldehyde form of our retinoid, and then this can be reduced by a reductase to form all-trans retinol. Okay? So following uptake, this beta-carotene can be cleaved and give rise to our retinol, right? So sometimes that's why they call this pre-vitamin A. So about 60 to 70% is going to be cleaved in the intestine. You can detect all-trans beta-carotene in the circulation, in the blood. And there are other tissues that express this monooxygenase BCM01. So it's likely being cleaved in other tissues as well. But some amount of this is circulating in the blood. But a good portion, 60 to 70%, is metabolized directly in the enterocyte and cleaved. Okay, so the rest is able to circulate in the blood to other tissues. And a lot of these do express this enzyme. It's possible, and there's speculation, that beta-carotene could have other effects beyond its metabolism into retinol or other retinoids, but most of the effects are thought to be from its metabolism into these vitamin A retinoid forms. Okay, so inside the cell and throughout our body as these are circulating, they're most commonly found bound to binding proteins, and there are a number of different binding proteins present. We won't talk about all of these, but this makes sense because it's a fat-soluble enzyme, so it's not very happy in aqueous environments. And so there are quite a few binding proteins that bind and mediate transport and sequestration of these fat-soluble molecules. So one of these that I mentioned was the cellular retinol binding protein, CRBP. And so there are a bunch of different family members. They're retinal specific, they're retinoic acid specific. binding proteins in their different isoforms of these as well. But for now, it's good to just recognize that there are both retinol and retinoid binding proteins inside the cell, and then we'll also talk about one that's present in the serum for transport. Okay. And so these serve a variety of important functions. One, they prevent non-specific retinoid metabolism. Okay, so they sequester these, they bind them and prevent them from being metabolized. In some cases, they can promote specific metabolism events by binding and mediating the transfer to select enzymes. And it's clear that these are really important because in addition to preventing these metabolism reactions, they also are able to maintain a large amount of the retinoids in the cytosol and decrease membrane concentrations of retinoids. And so this would, in the absence of these binding proteins, these retinoids, because they're fat soluble, would insert into our membranes, the cell membranes of these various tissues, which can have drastic effects on cell biology. Okay? And in addition, it's notable that the concentrations of these binding proteins are high. Okay, so they actually exceed the amount of retinoids we have in our cell, and this is just another reflection of the importance of these proteins in sequestering and binding these different retinoids. Okay? So we keep these binding proteins at very high concentrations to prevent any free retinoids from floating around, inserting in the membranes, or having these different metabolic reactions. Okay, and so this is an example of a crystal structure of the cellular retinol binding protein, CRBP. So it has this very classic beta barrel structure. It actually looks like a barrel, and inside, if you can see it in there, is the retinol molecule. Okay, so it's in the core of this barrel, right? So it's shielding it from all these potentially negative associations. And this is just looking down the barrel. It's kind of an oblong barrel, and inside is our retinol molecule. Okay, so upon entering into the enterocyte, we have retinol, we have our fatty acids, and retinol, similar to other fats we've studied, is often reesterified in the enterocyte. So just like when we take up triacylglycerol, we cleave off all the fatty acids, we take up the monoacylglycerol, and then we reattach all the fatty acids. So this is the same thing that happens to retinol. So the retinol ester is broken down in our gut, we take up retinol, and then we add back a fatty acid to form a retinol ester. Right? And this is mediated by two different enzymes, either an acyl-CoA, retinol acyltransferase, called ARAT, okay? And this is going to mediate transfer from a free acyl-CoA, which we get from ACSL, right? The acyl-CoA synthetase gives rise to an acyl-CoA, and then this enzyme can mediate the transfer directly onto our retinol to form the retinol ester. The other enzyme is this lecithin retinol acyltransferase, and this would similarly transfer fatty acid onto this retinol ester, but the fatty acid is coming from a phospholipid, like phosphatidylcholine or something like that. Okay, and both of these enzymes can form these retinol esters, but they're going to be getting the fatty acids from different sources. This one is coming from free acyl-CoA. This one is coming from a phospholipid, right? And so they're forming these retinol esters, which are going to be completely neutral and are going to end up being sequestered into chylomicrons. So these retinol esters, so similar, just if you're thinking about triacylglyceride or cholesterol esters, these then get packaged in the chylomicrons for secretion and transport through our body. Okay, so just summing this up, we have a few different forms of retinol coming in our diet. We have retinol esters, mainly coming from animal foods. And then we have beta carotene coming from fruits and vegetables. The retinol esters have to be metabolized, so the fatty acid is removed. So we have retinol plus a free fatty acid. These are trafficked into the cell. The beta carotene can be cleaved to form retinal. Often these are bound by our cellular retinol binding protein. The retinal can be reduced to form retinol, and then these can be modified again in the reesterified by LRAT or ARAT back to our retinal ester. Right? So just the same kind of idea. We take off the fatty acid, we move them into the cell, we stick the fatty acid back on using an acyl transferase. Okay. And then these retinal esters get packaged into chylomicrons, secreted out of the enterocyte into the lymph. Some amount is broken down and can be absorbed into the adipose, but the large majority will be present in chylomicron remnants and be taken up into the liver, where the majority of the vitamin A metabolism happens. Okay. So the liver is going to be the primary vitamin A storage tissue. They're going to have uptake of the chylomicron remnants. We're going to have breakdown in the lysosome and release of our retinal esters. And then these released retinal esters have two fates in the liver cell. They can either be stored or they can be secreted for transport through the body. Right? And if they're stored, they're going to be stored in hepatic lipid droplets. So these are lipid droplets in the liver. So if you remember in adipocytes, they form these gigantic lipid droplets that are full of fats and cholesterols. In liver, they'll also contain retinal esters. Okay, so these are neutral molecules. They can either be in chylomicrons or they're going to be stored in these lipid droplets, these cytoplasmic organelles. Okay, and these are thought to bud from the ER, similar to the idea of the formation of chylomicrons, and that those form by budding into the lumen of the ER, and then they can be secreted. These are very similar in structure to chylomicrons. They have a neutral lipid core surrounded by a phospholipid surfactant, and they bud into the cytoplasm from the ER. So very similar idea, right? Yeah, and so these serve as a dynamic storage site for retinal esters in the liver, and they can be hydrolyzed and released if we need them in the rest of our body. So the other route for retinal esters is hydrolysis and transport of the retinol in complex with a binding protein in the blood. Okay, so retinal ester, it's either going to be stored in these lipid droplets or it's going to be hydrolyzed. We'll release the fatty acid, we'll have retinol, it'll go out and bind to a serum retinol binding protein and circulate in complex with transthyretin, another circulating protein. So this is what that looks like in a crystal structure. So here's transthyretin, it's a tetramer. And then we have this retinol binding protein on either end. So we have one at the C terminus and one at the N terminus. And retinol, similar to our cellular retinol binding protein where it had that beta barrel, retinol is kind of sequestered in the middle of this retinol binding protein. Okay. And so this is how vitamin A is transported and circulated through our blood. And this was similar to a lot of the other vitamins we've talked about as well, that they bind to specific proteins in the circulation for movement. Okay, so just to summarize the metabolism in the liver, we have the retinal esters, which were present in our chylomicrons, and our chylomicron remnants. The chylomicron remnants are taken up by our liver. They're hydrolyzed in the lysosome, and retinal esters are released. And the retinal ester is broken down by the hydrolyses in the lysosome. So we'll have retinol. This can be either re-esterified by LRAT or ARAT back to retinal ester for storage in droplets. This can be hydrolyzed again back to retinol, and that can be then secreted and bound to retinol binding protein together with transthyretin for transport in the blood. Does that all make sense? Okay, so there are a few different forms of retinoids to be aware of. As I mentioned, these differ in the oxidation state of their polar head group. So we have retinol, so the alcohol, retinol. Retinal would be the aldehyde, and retinoic acid, right, the acid form. Okay? And usually when we think about the activities of retinoids in the body, we think a lot about retinoic acid. So this is the form of vitamin A which is going to influence gene expression. And we'll also talk about a specialized role for retinal as a chromophore in our eye. Okay? And so there are two enzymes which mediate the conversion of these different retinoids. We have retinol dehydrogenase, which converts retinol to retinal. And then we have retinal dehydrogenase, which converts retinal to retinoic acid. Okay? So these are the two main forms we're going to think about in terms of activity. Retinoic acid affecting gene expression, and retinal being having a specialized role in vision. Okay. So retinoic acid, abbreviated RA, activates specific transcription factors, which are referred to as retinoid receptors. And these come in a few different flavors. They're all members of nuclear hormone receptor ligand-inducible transcription factors. So this is similar to what we talked about in last class about vitamin D. So vitamin D was another vitamin which acts through these nuclear hormone receptors, right? And so there are different types of receptors. There are 3 retinoic acid receptors, abbreviated with this RAR nomenclature. So RAR alpha, RAR beta, RAR gamma. And then it can also bind to the PPAR beta and delta family of receptors. Okay? So these are going to be receptors for retinoic acid. Okay? And then the other receptor, this is for a specialized cis version of the retinoid, is retinoid X receptor. So RX, R alpha, beta, and gamma. Okay? So if you remember from vitamin D bound the vitamin D receptor, and then that vitamin D receptor heteroligomerized with those RXR receptors, right? And so these RXR receptors are kind of promiscuous. They bind to several different receptors which monitor and modulate gene expression in response to different metabolites. Okay, so there are different receptors which bind retinoid X receptors that are responsive to cholesterol or to vitamin D or vitamin A or other signals. So these RXRs are kind of a promiscuous receptor. So these receptors form these heterodimers. So we'll have heterodimers of RAR RXR or PPAR RXR. And these are going to be the active heterodimeric form when bound to ligands. So vitamin D, right, it was vitamin D receptor RXR was the active heterodimer. Okay. So these are going to bind to particular response elements. So these are retinoic acid response elements, RAREs. And these are in DNA. And in the absence of a ligand, these receptors actually heteroligomerize and repress the transcription of these genes that have these RARs, RAREs. But in the presence of the ligand, so in the presence of retinoic acid, it's going to activate these receptors and relieve the suppression and increase expression from these sites. Okay, we'll walk through that. So in the presence of a ligand, these receptors enhance transcription. Okay. So there are a few different forms of retinoic acid and retinoids which influence gene expression. So one is going to be this all-trans retinoic acid. This is what we've been abbreviating as RA. And this is the primary regulator of gene expression. It's the one that binds those RARs in the PPAR. Okay, so this is the most prevalent form in vivo. It's going to bind those nuclear RARs at a PPAR when this is sufficient to act as a transcription factor. So binding of all-trans retinoic acid to RAR, which will be in complex to the RXR, is sufficient to act as a transcription factor. The other form is this 9-cis retinoic acid. So it has a cis structure. And this is the form of retinoic acid which binds to the RXR. Okay, and this is that promiscuous receptor which heteroligomerizes with a variety of different nuclear hormone receptors. So binding of this 9-cis retinoic acid to the RXR is insufficient alone to influence gene expression, okay? Whereas binding of this guy to the RAR is sufficient. Okay? But binding of both seems to have more of an additive or synergistic effect. Okay, so in the absence of a ligand, the receptors are going to, you'll have a heteroligomerization of the receptors. They're just represented here as these little cylinders. And these are going to bind to these specific areas of DNA, in this case these RAREs, the retinoic acid response elements. And they're going to inhibit gene expression from these sites. And they do this by regulating the compaction of the DNA. So they recruit co-repressors, which are going to affect the histone packing of the DNA. Okay? So this results in increased packing around the histones and decreased gene expression from these target genes. Okay? So this is mediated by the co-repressors that are recruited by these heteroligomerized receptors. So this is in the absence of a ligand. In the presence of a ligand, so in the presence of our all-trans retinoic acid, this is going to bind to our RAR, which is present in this heteroligomer, and it's going to recruit a different set of coactivators. Okay? And there's a kind of a two-step phase that happens here. This first step is de-repression, so we get, again, kind of the alteration of the histone packing of the DNA. So here it's really packed into these, wrapped around these histones. Here we alleviate the packing of the DNA, allowing it to increase expression. And then it recruits a second set of factors, these mediators, which recruit RNA polymerase and other transcription machinery to increase the gene expression from these genes present in this DNA. Okay. Okay, so we have our transthyretin retinol binding protein bound to retinol, circulating in the serum. Retinol is taken up into the cell. This can be bound by our retinol binding protein. This can be converted by these dehydrogenases in a sequential manner. We have retinol dehydrogenase converting it to retinal, and then retinal dehydrogenase converting it to retinoic acid. And this is our ligand. So it binds to RAR, and then it influences gene expression in these cells by binding those RAREs. Is this pretty clear? Okay. And this is very similar to the vitamin D. Also has similar effects, right? Through nuclear hormone receptors. Okay, so with regards to our vision and why retinoids are particularly important in our vision, there are a few different forms to be aware of. This 11-cis retinal is going to be the star of the show. And then we also have our all-trans retinal and our storage form, our retinal esters conjugated with a fatty acid. Okay, so we have shown here is just a schematic of what the cells in our eye look like. So here would be the capillaries, we have pigment epithelium, and then underneath the various cells that detect our light. So we have rods, we have cones, and then some interspersed mular cells, which are support cells. And so rhodopsin is an important protein in the detection of light. That's why we can see. This is a membrane protein in photoreceptor cells that mediates the sensing of light. And it's able to do this because it coordinates this 11-cis retinal as its chromophore. Okay? So 11-cis retinal is what's actually detecting the light. So absorption of a photon by 11-cis retinal is going to trigger a cascade of reactions that end up resulting in hyperpolarization of the membrane of the cell, which can then be transmitted to secondary neurons, which then communicate with the visual cortex of our brain and the occipital lobe. Okay? So the 11-cis retinal serves a really important function in actually detecting the light and being the mediator or initiator of this cascade of events, allowing the rods or the cones to communicate with our neurons and communicate that we've seen the light to the respective visual cortex of our brain. So this is just kind of another representation of that. This is a representation again of a rod. So the rods are detecting black and white images, light, not color. And in the outer segment of the rod, there are these discs which contain our rhodopsin. Okay? And our rhodopsin is a polytopic membrane protein which is coordinating 11-cis retinal right here in its core. Okay? So rhodopsin contains retinal, or 11-cis retinal, together with the protein opsin. We call that rhodopsin. Okay? And what's important is this conversion from the 11-cis isomer, 11-cis retinal, to our all-trans isomer. Okay? So in response to light, we have this conversion. And this conversion results in a conformational change in the enzyme in rhodopsin, which mediates this cascade of events. So let's just walk through this. We have a photon of light, and we have our rhodopsin, which is coordinating our 11-cis retinal. So in response to the light absorption, 11-cis retinal is converted into all-trans retinal, right? And this conversion. And this is a pretty big structural change in that retinal molecule. And this is just represented here as R star. This can then bind to transducin and coordinate the exchange of GTP. So it binds transducin, and there's an exchange of GTP with GDP exiting. Okay, transducin bound to GTP can then activate an enzyme called phosphodiesterase, converting it from an inactive form to an active form. Okay, so again, kind of this sequence of events where a single photon is starting to amplify the signal. Right? So we have a single photon here. Maybe we have multiple transducins exchanging GTP, and then we have activation of an enzyme, which is going to be a big amplification in the signal. Okay? So the active phosphodiesterase cleaves a bond, converting cyclic GMP to GMP. Okay? And this is important because cyclic GMP regulates sodium channels in that rod, which in the presence of high cGMP, those sodium channels are open. Okay, so the cell is going to be depolarized, and it's going to be secreting neurotransmitters like glutamate and controlling those secondary neurons, which are firing to our visual cortex. So in response to light, we have all these events happening, all culminating in these sodium channels closing, and our cell becomes hyperpolarized, and it shuts off transmission to these second-order neurons. Okay? Is that clear? We have light. So what's really cool about this is we're converting light into a chemical signal, right, that we can actually interpret.
So you say, like, in response to light, the neurons are, like, deactivated?
So the firing from from the rods to the second order neurons goes down because these will be hyperpolarized. Yeah. Good. And so, of course, now that we have this conversion, we have rhodopsin going from 11-cis retinal to all-trans retinal. It wouldn't be able to detect light anymore because it's in the all-trans form. So we have to have a recycling step to convert this back, to get back to rhodopsin plus this 11-cis retinal, so it can detect another photon of light, right? And so this actually happens through an exchange of these retinoids between the different cells in the eye, between the photoreceptor cell, which we were talking about, the rod, right, the rod cell, and cells in the pigment epithelium, so retinal pigment epithelial cells. So we have this event. We have rhodopsin, which is opsin plus our 11-cis retinal. There's light, converting it to an all-trans retinal plus opsin. Okay, so opsin can be recycled, just needs more 11-cis retinal. The all-trans retinal is converted to all-trans retinol by a dehydrogenase, or retinal dehydrogenase. And then this can leave the cell, actually leaves the photoreceptor, and goes over to the pigment epithelial cell for recycling. And a few things can happen in the pigment epithelium. So the all-trans retinol is converted into the retinol ester, and this can perhaps just be stored as the retinol ester in lipid droplets in these cells, and this happens through the actions, most likely, of LRAT. This can then, the all-trans retinol ester is then hydrolyzed, and during this hydrolysis step, it's hydrolyzed into conformation of 11-cis retinol. And this can cycle back and forth between an 11-cis retinol ester. Okay, and this 11-cis retinol can then be converted into 11-cis retinal by a dehydrogenase and transferred back to our photoreceptors for reincorporation with opsin into rhodopsin proteins. So we just have a series of changes in the conformation of that retinal, right? From an all-trans back to an all-cis. And so we have a couple different steps of the dehydrogenase in a passage between the cells to get back to 11-cis retinal, which can serve as the chromophore. Okay, so kind of summarizing the key points regarding retinoid metabolism and vision. We have retinal pigment epithelia. These are important cells for the recycling of that 11-cis retinal. It contains stores of all trans retinal esters. So these are going to serve as sources for that 11-cis retinal. The RPEs convert the all trans retinal esters into 11-cis retinal in a series of hydrolysis and dehydrogenation steps, which can then be supplied to the photoreceptors for incorporation into the rhodopsin. So 11-cis retinal binds to opsin, giving rise to rhodopsin, and it serves as the important chromophore within this protein, detecting the light. So in response to light, 11-cis retinal is converted into all trans retinol, and then it can be transferred back to the RPEs for storage or for regeneration of our 11-cis retinal. Does that make sense? So in terms of deficiencies, vitamin A deficiency is still prevalent in developing countries. It's not that prevalent in developed countries, so it still can happen. That's the case with most of these vitamins that we've been discussing because it's present in a lot of the foods we eat, which are pretty widely available, and it's also heavily fortified in our cereals and other foods. So cases in probably the United States where we get this might not necessarily be to deprivation. It might be something like pancreatic insufficiency or impairments in fat absorption rather than a deficiency. So in the lack of vitamin A, you're going to get things like night blindness. That will be pretty rapid, which makes a lot of sense. You can also get more severe eye conditions, which can lead to blindness. So we mainly focused on retinoic acid's importance in the eye, mainly as eye health and as the chromophore in rhodopsin, but also plays a lot of other important roles. We won't go into as much detail with these. It plays important roles in skin, and it plays important roles in reproduction, most notably in pattern development during embryogenesis and during regeneration, during healing processes. And so this is a particularly striking example is kind of illustrated here in this classic experiment. So I think this is kind of a neat field. So there are some scientists studying limb regeneration, and they study this in amphibians. So of course we can't regenerate our limb if you cut it off, but amphibians can. And so they do this either in amphibians like newts or in like frogs. They actually cut the limb off, and then they can observe it growing back over the course of maybe 2 months or 3 months or something like that. And so normally it's able to actually reform a normal limb over that time. And retinoids are critical to this process. So the limb bud express RARs, the retinoic acid receptors, and these seem to be important for the regeneration of the limb. And you can actually apply retinoic acid to the limb bud in different places and result in really weird things. If you apply retinoic acid aberrantly, you can get like double limbs or extra shoulders in the limb. Okay, so retinoic acid is really important, but it's important to have the correct amount of it at the right places at the right time for proper generation of the limb. Okay, which is really neat. So this is kind of a cool area of science in general, just the idea of limb regeneration or regeneration in general. You can imagine the possibilities if we were able to understand that process in more detail. It's also used in skin, so you probably see topically. I think it's pretty safe. So you'll see a lot of like anti-aging lotions and creams will have retinol in it or other retinoids in them for topical application. And also anti-acne medicines for topical application have a lot of retinoids. For very severe forms of acne, there are systemic forms of retinoids that are quite effective in treating acne. So these are probably the most widely known version of this would be Accutane. And these can be quite effective. So these are taken systemically, so they're going to be throughout the body. But they have a lot of serious side effects. One of them includes birth defects and or death of the fetus, which might be consistent with what we just saw in the amphibians, in that retinoic acid and retinoic acid receptors are playing this really important role in embryogenesis as well as pattern formation. So whenever you take these types of medicines, it's really important that the individuals taking these usually are on birth control or at least are heavily suggested not to get pregnant. Especially, this is a big problem in the first trimester of pregnancy. It's not a problem taking with the creams, like the topically applied lotions and things like that. Not much of it gets into the systemic circulation. It's also been associated, and this isn't as well understood, with depression and suicide in adolescents. So it has a lot of potential side effects. But the most serious, I would say, is these. Well, they're both serious side effects. So these are pretty devastating. Okay, so I think we made it through vitamin A. This is the end of this lecture. Before you guys leave, I wanted to remind you about, so I'm going to stay around and review some if you guys want, but this isn't going to be comprehensive. We just don't have time, of course, so I'm just going to touch on some, just maybe see this as some reminders about what we did during the first 6 lectures of the class, and some reminders of things that I think were important topics from those classes. So I won't be able to cover everything from all those classes. But these could be things that maybe you want to go back and study a little bit because they'll probably come up on the exam. Okay, so if you remember from way back in lecture 1, we went through the basic anatomy and physiology of the gastrointestinal tract. So we went through each of these different organs and stressed some of the anatomical specializations of the cells in the different organs and the functions of the different organs, right? So these have come up again and again, and we probably are fairly familiar with them by this point. So we talked about the stomach, we talked about the different cell types in the stomach, the mucus cells, the parietal cells, the chief cells, what types of enzymes they secrete, and their functions in the stomach. We had a particular focus on acidification of the stomach by parietal cells. And so everybody remembers this guy, which was a little confusing. But it was just showing that we have these different transporters present in our parietal cell, which allow for secretion of protons and chloride ions to form hydrogen chloride. And so basically we have gases like CO2 and water. We have our upstream enzyme carbonic anhydrase producing the proton and bicarbonate. Right? And that proton can then be pumped out through an ATP-dependent primary active transport step in exchange for potassium. And then our bicarbonate actually exchanges for chloride in the blood. Chloride comes into the cell and then goes out of the cell by facilitated diffusion, giving rise to our acidified stomach. Right? So there are a few different transporters to remember, and then one enzyme, carbonic anhydrase, to remember with this. We had a few different regulatory steps here, different cell types that would influence this process. So we had the G cells secreting gastrin. We had the vagus nerve innervating the stomach secreting acetylcholine. And then we had enterochromaffin-like cells secreting histamine, right? And so these are all positive regulators of acidification of the stomach. And one of the big regulators that we focused on was the release by enterochromaffin-like cells of histamine, right, onto this H2 receptor on the parietal cell. So we talked about the different cascades that resulted in increased release of acid. We also talked about the synthesis of histamine from histidine in the enterochromaffin-like cell. And so one way that gastrin in the vagus nerve, by releasing acetylcholine, increase acidification is they increase the production of histamine, allowing the enterochromaffin-like cell to secrete more histamine. Right? So we also talked about negative regulators, somatostatin, but we didn't go into as much detail there. Okay, so there were a bunch of other organs that we discussed. We discussed the pancreas, which has a few different cell types. And remember that the pancreas is playing endocrine and exocrine functions. So in terms of the endocrine functions, which were probably covered more in Hasek's lectures, we have the alpha and beta cells present in the pancreatic islet, which are secreting things like glucagon and insulin into the blood. And then we have the acinar cells present in these bundles secreting digestive enzymes into the pancreatic duct, which then will go into the common bile duct and into our duodenum, right? So we spent a lot of time on small intestine and enterocytes. So just remember the anatomical specialization of the small intestine involves absorption, which makes sense for its function. And so a big part of the anatomical specialization is expansion of surface area of the intestine, both through villi, which are going to be lined with our enterocytes. And then on our enterocytes are the microvilli, right, which are membrane projections. So these are macro projections, lots of cells covering the surface. And then we have our microvilli or brush border, which are little micro membrane projections off of single cells. Right? On the surface are going to be all kinds of our enzymes involved in breakdown, as well as receptors involved in absorption of nutrients.
Did you say that the acinar cells are exocrine or endocrine?
Exocrine. Yes. Okay, and then another important topic from this was the enterohepatic circulation, which just refers to the hepatic portal vein, which brings material absorbed from the small intestine back to the liver first. So the first site is going to be the liver, and so things like bile acids are going to be reabsorbed into the liver for recycling so that they can be resecreted via the common bile duct. And this was really important for bile acids and bile salts to enable recycling of those molecules. So remember that bile acids are those cholesterol-derived molecules important in forming mixed dietary micelles and emulsifying the fats for digestion. Okay. In lecture 2, we went over a bunch of different transport mechanisms, and we probably touched on these a lot as we talked about absorption and movement of molecules throughout the body, uptake into different tissues. But just refresh your memory a little bit on these. We talked about bulk transport mechanisms, so diffusion, down concentration gradients. We talked about facilitated diffusion, which is both of these aren't going to require any energy, right? Because they're going down their concentration gradient, facilitated diffusion requiring a protein mediator. And then we talked about energy coupled transport mechanisms, including primary active and secondary active, and the bulk transport mechanisms like endocytosis and exocytosis. And so these came up a lot when we think about chylomicron remnant endocytosis or different receptors being endocytosed, as well as the secretion of a variety of hormones. These all go through exocytosis, right? So this would be the snare-mediated fusion of vesicle membranes, ATP-dependent. Endocytosis would be the clathrin and dynamin pinching off ATP-dependent steps, right? So requiring some energy. We talked about a variety of different barriers to transport, and so depending on what type of nutrient or molecule you are, you might face different barriers, whether you're a fat-soluble vitamin or a glucose molecule, right? You're going to have different barriers to your transport. One of these was the unstirred water layer, which is going to primarily be a barrier for fats or fat-soluble materials. And the unstirred water layer is just a thin layer of water immediately proximal to the brush border, and it's just created because all the proteins on the brush border of the enterocyte have lots of glycans, which are extremely hydrophilic. They're binding lots of water, and so you get this kind of shell of water, which isn't a lot of movement or diffusion of that water, and so we call it the unstirred water layer, and that's functioning as a barrier. So we have the incorporation or breakdown of fats into incorporation into these mixed micelles with the bile salts, allowing them to cross this unstirred water layer for uptake into the enterocyte. We have these variety of transport mechanisms, which we touched on, the passive transport mechanisms and active transport mechanisms requiring energy. So anytime something is going against its concentration gradient, just remember that that'll be active. If it's going down its concentration gradient, that'll be passive. Okay. And the two types of active were ATP driven, which is going to be primary active, and that's if it's directly at the transporter. So for example, like the proton pump and the acidification of the stomach, or the sodium-potassium pump in our enterocytes. Those are examples of primary active. And then we have secondary active, in which primary active is establishing a gradient, and then you have symport or some sort of coupled transport to allow for movement of a molecule up its concentration gradient. Okay? So secondary active is utilizing a concentration gradient of another molecule to drive the movement of this up its concentration gradient. So an example of that would be the SGLT1 glucose transporter. Right? So it uses sodium going down its concentration gradient to drive glucose up its concentration gradient. There's no direct ATP being hydrolyzed by that transporter, but it's coupling movement of sodium down its concentration gradient, right? So in lectures 3 and 4, we moved on to talk about the digestion, breakdown, and absorption of sugars. These came in a variety of different flavors. We had glucose, fructose, and galactose, and some of these could be joined together to form things like sucrose, which was joining of glucose and fructose, or lactose, which is galactose and glucose together, right? Similar disaccharides. And then we have polysaccharides, which could also come in different flavors. So amylopectin or glycogen, which is going to have a branched glucose chain, or amylose, which is the long string of glucose polymers. And these are being joined by glycosidic bonds, right? They're joining these guys. And the structure of the glycosidic bond was important for whether or not we could actually break down the various types of glucose polymers, right? So our body can't break down all glycosidic bonds. Okay. We also talked about the Maillard reaction, which is important in cooking. And this is important to remember that this is a non-enzymatic reaction. So we call this glycation. And this is opposed to regulated enzymatic reactions that happen in our body called glycosylation, right? And so things, when our proteins transit through the endoplasmic reticulum and make it to the outside of our cell, they get glycosylated, and that's a regulated enzymatic process. And so this is one example of glycation that happens in our body, and it was a useful metric for how well, it's used as a useful metric for how well diabetics are controlling their blood glucose, right? And this is because hemoglobin is this really long-lived protein, right? And so over time, it'll reflect the glucose levels, or maintenance of glucose concentrations in the blood. So they call this the hemoglobin A1C test, and they analyze the amount of glycated hemoglobin. So if you just measure glucose, that's going to be acute measures, right? And it'll change really frequently depending on if you've eaten or in what state your body's in. But glycation will reflect your glucose control over the last few months. Right? Okay, so we had a variety of enzymes involved in the breakdown of sugars coming into our body. So one of the, we have sugars coming in the form of those disaccharides and free monosaccharides, and in these larger polymers like amylose or amylopectin or glycogen from animal cells. And so amylase, secreted by the pancreas, was one of those enzymes that was important for the breakdown of glucose polymers. And it could cut within the polymer to produce smaller disaccharides or trisaccharides, right? And this is because it binds sort of an active site of 5 glucose residues and cuts in this residue. This is kind of an endo reaction. But it can't cut these branch points, right? So when it continues to cleave this, eventually we're going to get to what I refer to as alpha-limit dextrins, which are unable to be further hydrolyzed by amylase. So we require additional sets of enzymes to digest these. So remember that amylase is being secreted by the pancreas. It's a soluble enzyme. And then we have a variety of saccharinases that are attached to the enterocyte brush border. Right? So they have transmembrane domains, and they're stuck on the brush border of the enterocyte. And these allow us to finish digesting these alpha-limit dextrins. So we have our alpha-limit dextrin of glucose that has these various bonds, alpha-1,4, and then those alpha-1,6 branch points. So we have glucoamylase, and so it's an exo cutter, so it's going to chew back. So it takes off a glucose, and then we hit our alpha-1,6 branch point. This is cleaved by alpha-dextrinase, also known as isomaltase. And then this can be finished being digested and hydrolyzed by glucoamylase and maltase. Okay? Makes sense. And these enzymes had a variety of different activities, so there's a really confusing slide in which it says the different activities, but I think understanding this basic cascade is sufficient. So glucose and fructose and monosaccharides can then be absorbed. So anything that's present in a disaccharide or bigger isn't going to be absorbed by our enterocytes. And we have a variety of different transporters present on the enterocyte brush border. These included things like SGLT1, which is our good example of secondary active transport. So we have that sodium potassium ATPase pumping sodium out. That's our active transport. And then sodium and glucose can flow in through SGLT1. And so this would be primarily acting under low glucose conditions to move glucose up its concentration gradient. Right? And then it can then move out of the enterocyte via GLUT2 on the basolateral membrane into the blood. Right? And then our other player, we have GLUT5, who is mediating the uptake of fructose, and then we have GLUT2 also present in vesicles. Okay? And these GLUT2 present in vesicles allows our body to adapt to conditions of high sugar and mediate continual uptake of glucose under these high sugar conditions because it has a high Vmax. And so GLUT2 under high sugar conditions is trafficked to the apical membrane. SGLT1 is still working, but it's becomes saturated very quickly, right? It has a low Vmax. So under these high conditions, GLUT2 can mediate uptake of glucose and fructose, and then it can go down its concentration gradient into the blood. So this is just a representation, again, of those properties of the Vmax. So the Vmax would be the saturation concentration of glucose at which there's maximal velocity of transport. So GLUT2, you can see, has this really high Vmax. So it's going to continue to be able to increase its transport of glucose, even if we increase the concentration of glucose to really high levels, whereas SGLT1 becomes saturated, right, because of that lower Vmax. Okay, following the small intestine, we reach our large intestine, and one of the big points about the large intestine was that it's home to a host of microbiota, right? And these contribute to the breakdown of some material which makes it to the large intestine. So prebiotics are non-digestible food that are promoting the growth of our intestinal microbiota, whereas probiotics are live organisms that we can use to that can make it to our large intestine and colonize our large intestine, perhaps changing the microbial communities in our large intestine. And one thing we learned was that if we didn't have enough lactase, for example, we'd get sugars like lactose making it down to the microbiota, and this could produce gas and other uncomfortable side effects because these bacteria organisms would be metabolizing that lactose. Okay, so in lectures 5 and 6, we discussed the breakdown and absorption of lipids. And there are a number of players present here. Most of this is going to happen in the small intestine, right? So there's a small amount of digestion that's happening in the stomach because we have gastric lipase present, but it's only cleaving some medium chain fatty acids from some of these triglycerols. And so very little triglycerol digestion is happening in the stomach. These are still present in pretty large fat globules, right? When we get to the small intestine, we have the secretion of bile acids, which allows us to take those huge fat globules and emulsify them and form smaller globules, right? And emulsified fat. And these are smaller fat globules which can then be more easily accessed by the different enzymes for the fat digestion and broken down, because we increase the surface area. So we go from like a giant fat globule to lots of little fat globules. And then we discussed a number of different enzymes in their substrates and their products, right? So we have pancreatic lipase, which is working together with colipase to break down triacylglycerol, right, to monoacylglycerol. We have carboxyluster lipase, which is breaking down cholesterol esters. We also learned today that it breaks down retinol esters, right? Leaves retinol esters and releases retinol and a fatty acid. So this guy is working overtime. And then we had phospholipase A2, which is digesting some of the phospholipids. And we learned this is taking a phospholipid, which has 2 acyl chains, and removing that SN2 middle fatty acid. And it also serves roles in our cells, right? It removes the SN2 arachidonic acid for the formation of eicosanoids, right? So we have phospholipase A2 present in our stomach. It's also present, in our small intestine. It's also present inside cells for eicosanoid production. And it serves the same function in cleaving that SN2 fatty acid. So all of these enzymes are digesting these lipids, and then we get finally this mixed micelle of digested lipid products together with our bile acids, which can cross the unstirred water layer. Okay, we discussed some of the essential fatty acids. We have 2, linoleic acid and linolenic acid. I'm not worried about you memorizing every single one of these steps. One of the most important lipids that's generated from our linoleic acid is arachidonic acid. So we spent a little bit of time focusing on arachidonic acid because it's the precursor for eicosanoids. Arachidonic acid cleaved off the SN2 by phospholipase base A2, and then that can be used to produce eicosanoids, which are lipid signaling molecules. Right? Okay, so following uptake, so we have uptake at the enterocyte apical membrane. We have things like CD36 and other transporters. Also have cholesterol efflux proteins at the apical membrane. And then we have reesterification and reformation of triglycerides. And we've seen this again and again, that we have breakdown, absorption, remaking it. It seems really wasteful, but this happens all over the place in our digestive system and circulation. So when we talk about reesterification, we mean the reformation of these ester bonds on this glycerol backbone to reform our triglyceride. So we have 3 ester bonds, 3 fatty acids, which are glycerol backbone. We could also say that for our vitamin A, our retinol ester, it's hydrolyzed by carboxyl ester lipase, it's taken up, and then it's re-esterified, right? We reform that retinol ester. And this occurs through a series of enzymes. One of the important ones was acyl-CoA synthetase, and so this takes a free fatty acid and conjugates it together with coenzyme A in an ATP-dependent reaction. And so anytime you have an acyl transferase reaction, anytime you're moving an acyl chain, it's probably involving an acyl-CoA synthetase to activate that fatty acid first, anytime you are attaching a fatty acid. And so in the monoacyl glycerol pathway, this is what's primarily occurring in our enterocytes, because we have a lot of MAG, monoacyl glycerol, coming from our diet, right? We have a ton of MAG getting in their enterocytes, and these are reesterified through these monoacyl glycerol acyl transferase step. So we take our acyl-CoA, formed by our acyl-CoA synthetase, we attach it to form DAG, and then we attach another acyl-CoA, take another acyl-CoA and attach another acyl chain to form our TAG. Right? The other pathway, this isn't as prevalent in enterocytes because these have a ton of MAG, but you can also make triacylglycerol from glycerol 3-phosphate, and this pathway is more active in places like adipose tissue. You'll have a series of enzymes that mediate the attachment of these acyl chains to glycerol 3-phosphate, eventually coming to DAG, which can be esterified to TAG by DGAG. Okay. And then we have our buildup of our chylomicrons. The important guys to remember here, so this is all taking place at the endoplasmic reticulum. One of the important proteins is the microsomal triglyceride transfer protein, which is putting triacylglyceride together with our apolipoprotein B. Okay, so it's a big player in these chylomicrons is apolipoprotein B and getting the triglyceride. And this is surrounded by a phospholipid. So this gives rise to our pre-chylomicron, which then can move through the secretory pathway through the Golgi and be secreted, gaining a few more other apolipoproteins. But the apolipoprotein B, MTP, and the TAG are important steps in this process. Okay, I think that wraps up 1 through 6. And I'm happy to take